RNA polymerase is the molecular machine responsible for transcribing DNA into RNA. This enzyme plays a central role in gene expression, acting as the bridge between the genetic information stored in DNA and the functional RNA molecules needed by cells. Throughout this video, we'll explore how this remarkable enzyme works to create RNA from a DNA template. RNA polymerase is a large enzyme complex that synthesizes RNA from a DNA template. Its primary function is to catalyze the transcription process, which occurs in all living organisms. In eukaryotes, you'll find this enzyme working in the nucleus, while in prokaryotes, it operates in the cytoplasm. The products created by RNA polymerase include messenger RNA, ribosomal RNA, and transfer RNA, all essential for protein synthesis and cellular function. The complexity of RNA polymerase differs between prokaryotes and eukaryotes. Prokaryotes have a simpler system with just a single RNA polymerase that transcribes all genes. This enzyme has a relatively simple structure. In contrast, eukaryotes have evolved a more complex system with 3 distinct RNA polymerases: RNA polymerase I, II, and III. Each of these has specialized functions and a more complex structure to handle the sophisticated gene regulation needs of eukaryotic cells. Let's examine the 3 types of eukaryotic RNA polymerases more closely. RNA polymerase I transcribes most ribosomal RNA genes, which are crucial for making ribosomes. RNA polymerase II is responsible for transcribing messenger RNA and most non-coding RNAs. This is the polymerase that transcribes protein-coding genes. Finally, RNA polymerase III transcribes transfer RNA and 5S ribosomal RNA genes. This division of labor allows eukaryotic cells to regulate gene expression with greater precision. RNA polymerase has a sophisticated structure that enables its function. The core enzyme contains multiple protein subunits that form the catalytic center for RNA synthesis. Within this structure is an active site, a channel where the DNA template enters and the newly formed RNA transcript exits. A key structural element called the bridge helix runs through the center of the enzyme, connecting its major domains and playing a critical role in the catalytic mechanism. Transcription occurs in 3 distinct phases. The first phase is initiation, where RNA polymerase binds to the promoter region and unwinds the DNA double helix. Next comes elongation, during which the enzyme moves along the DNA strand, synthesizing RNA as it goes. Finally, termination occurs when RNA polymerase releases the completed RNA transcript. Each of these phases involves specific molecular events and regulatory mechanisms. The initiation phase begins when RNA polymerase recognizes and binds to a specific DNA sequence called the promoter region. Once bound, the enzyme causes the DNA double helix to unwind, creating what's called a transcription bubble. This exposes the template strand of DNA. The enzyme then adds the first few nucleotides to begin RNA synthesis. This is a critical step because it determines where transcription will start and which genes will be expressed. During elongation, RNA polymerase moves along the DNA template strand in the 3' to 5' direction. As it moves, it synthesizes a new RNA strand that grows in the 5' to 3' direction by adding nucleotides one at a time. The enzyme ensures that complementary RNA nucleotides pair correctly with the DNA template bases. This process proceeds at approximately 40 nucleotides per second, demonstrating the remarkable efficiency of this molecular machine. Termination mechanisms differ between prokaryotes and eukaryotes. In prokaryotes, termination can be row-dependent, which uses a protein called Rho, or row-independent, which relies on hairpin structures in the RNA that cause the transcript to dissociate from the template. In eukaryotes, the process involves cleavage and polyadenylation signals. The transcript is cleaved and released, while the polymerase continues briefly before dissociating from the DNA. These mechanisms ensure that transcription stops at the appropriate location. To summarize, RNA polymerase is an essential enzyme fundamental to gene expression in all organisms. The transcription process it carries out involves three carefully orchestrated phases: initiation, elongation, and termination. This enzyme demonstrates high specificity, recognizing particular promoter sequences and synthesizing accurate RNA transcripts. In eukaryotes, multiple types of RNA polymerase exist, each specialized to transcribe different classes of genes. Understanding RNA polymerase is crucial for comprehending how genetic information flows from DNA to RNA to proteins in living cells.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.